The title of this project is an analysis of polysialic acid, its contributions to non-small cell lung cancer, and the NK cell and killer T cell response. Cancer is one of the largest diseases in the world and takes thousands of lives every year. And so far, we haven't found a cure to it, but this project will help us understand cancer and the immune response to it. This brings up the research question. How does polysialic acid contribute to the growth of cancer and the subsequent immune response to it? So there's two types of immune responses. There's the innate immune response, and then there's the acquired immune response. The innate immune response is something that we already know. It's something that's already in our DNA. While the acquired immune response is something we learn over time about when we face different types of pathogens. Today we'll be discussing about the innate immune response. So all cells have something called the major histocompatibility complex. See, but cancer cells and pathogens do not have the same kind of protein. So what happens is these B cells go around in the bloodstream, and when they find a pathogen or a cancer cell that doesn't secrete the same type of protein, it alerts it. And the way it alerts it is it secretes this something called an antibody, and an antibody is a Y-shaped protein. And this Y-shaped protein binds to these specific receptors on the cell surface. It's something like a key and lock method, where there's only a specific type of lock that will work for this specific type of key. And then from there, those antibodies will alert the dendritic cells. Well, then the dendritic cells will alert the killer cells, and those killer cells will come in and try to kill the cancer. Now, what is polysialic acid? Polysialic acid is a post-translational module. So what happens is all proteins are made out of amino acids, and amino acids are subsequently made up of a universal structure or a backbone. And what polysialic acid does is after the amino acid is already created, it adds another group or another series of elements to either the C or N terminus. In polysialic's case, it adds a glycan or a polysaccharide to polysaccharide to the C terminus of the amino acid. And then from there, that amino acid dramatically changes its chemical and physical properties. Chemically, the polysialic acid creates an acidic sugar, essentially, and it becomes very anionic or negatively charged. And then physically, that same polysialic acid creates a glycocalyx or a bubble around that cell. So let's see a picture of this. This is an electron graph of the glycocalyx. See, this is all the intercellular, and that's inside the cell. On the edges, we have the lipid bilayer, or the natural protection or barrier around the cell. And all this black stuff, that's all the glycocalyx, or what the polysialic acid creates. And why there's so many electrons here goes back to how it's so anionic or negatively charged. So what are its uses in NCAM? In NCAM is like the neurons and the nervous system in your body. See, it's not such a problem for your immune system because your immune system can't attack your nervous system. And what happens is this same polysialic acid or the same glycocalyx forms around these neurons and helps the cell-to-cell interactions. See, if there was no acidic bubble or the glycocalyx, these cells would not be repelled and they would clump together. And these neurons clumping together is very bad for the mind and the body because this creates several disorders like autism or bipolar disease. But what cancer we suspect is doing is it itself gets and mutates to get that same polysialic acid. So what the hypothesis is that if polysialic acid was found in cancer, the cancer would be more resistant to the immune response and would increase in cell viability. So one study done was by Dr. John Eden at the University of Louisville. He tested several strains, including of E. coli in a mammalian blood serum. See, E. coli is very different, where some species of E. coli or some strains have polysialic acid, while the others do not. This tests the effect polysialic acid has itself without cancer. And what the 5 different cell lines showed was cell lines that didn't have any polysialic acid, like 0064, went from 100 cells to 35 cells in 2 hours, and all the way down to just 3 cells surviving at 4 hours. While cell lines that did have polysialic acid, like RS21A, went from 100 cells to 116 to 190. This shows that the polysialic acid sort of protected itself because it showed that the statistical analysis, and then specifically the standard t-test, had a value of 14.58. And this shows that it was significant. And this shows that polysialic acid did actually indeed have an effect on how long these pathogens or E. coli survive. But E. coli and cancer are not the same thing. So we need to find out if cancer does have this polysialic acid, because now we know that polysialic acid is something that helps protect whatever it's on. This brings up the rationale. We want to see a pathway of cancer, and we're going to see if cancer actually uses polysialic acid. So the methodology was to grow out these cell lines, and these cell lines included 1792, 8549, and H1299. And these cells were all cancers, or they had oncogenes in them, while HBEC was the only cell line that didn't have any oncogenes and was a normal cell. We let them grow up, and then we would put Actane blue on them. Actane blue would stain and glow up if it found polysialic acid. So we wanted to find if cancer did have a direct correlation with polysialic acid. And these were the results. See, all these illuminated largely and became bright blue because of the Actane blue stain positive on the cancer cells because of the polysialic acid, while HBEC stayed the same because it had no polysialic acid. So what does this mean? This means that the hypothesis was tested positive because we found that polysialic acid is a cancer-specific trait and thus could be one of the reasons how cancer protects itself from the immune system, therefore supporting my hypothesis that it would increase cell viability and help cancer because it's one of the pathways that helps cancer protect itself. So what are the implications? We can use this in future research to find the immune response and some way find an antibody that can bind specifically to and make it through the polysialic acid so it can recognize the cell where the immune response can finally just kill the cell, and thus could be one of the reasons how we solve cancer. And the future research is I can do it in mice or in living organisms for better results. And this is Big Leon. Thank you for listening.